Hi guys, welcome to our podcast, Complete Combustion, where we talk about biology and chemistry and other sciencey things.
So today we're doing biological molecules, so who wants to start?
Shall I start? Okay,
You can if you want,
What elements are required for living processes? 4 key elements which make up biological molecules and are essential for all living processes are hydrogen, oxygen, carbon, and nitrogen. Inorganic ions also play an important role in living organisms.
Macronutrients are needed in small amounts, such as inorganic ions. What are some examples?
Magnesium, which is a constituent of chlorophyll and is essential for photosynthesis. Plants without magnesium in their soil, they can't make chlorophyll, and so the leaves are yellow, and that's called chlorosis.
Very nice. You've got iron, which is a constituent of hemoglobin, which transports oxygen inside red blood cells. Lack of iron in the human diet can lead to anemia.
We also have phosphates, which are used for making nucleic acids, so your DNA, RNA, and ATP. It's a constituent of phospholipids and is found in plasma membranes.
And then you also have calcium, which is a structural component of bones and teeth in mammals, and a component of plant cell walls, providing them strength.
Very nice. Micronutrients are needed in minute, which is trace amounts, only, and so some examples are zinc and copper ions. Definitions?
Okay, then there's also a couple definitions. So micronutrients are inorganic nutrients needed in a minute trace amounts. So copper and zinc, basically the same thing.
A compound could be anything that consists of more than one type of atom, like 2 or more elements joined together.
Inorganic compounds are compounds that do not contain carbon bonded to hydrogen, so for example water, carbon dioxide, and nitrogen gas.
And then macronutrients are inorganic ions needed in small amounts, so magnesium, iron, phosphate, and calcium.
An atom is something that contains electrons, protons, and neutrons.
Whereas elements, they only consist only of one type of atom.
And then you have a molecule, which is 2 or more atoms joined together, and these can either be the same or different types of atoms.
Yeah, cool. Organic compounds are compounds that are based on carbon and contain hydrogen bonded to carbon, such as carbohydrates, proteins, and lipids.
So, monomers and polymers. So carbohydrates and proteins can form very large polymers. So what are polymers? Polymers are made up of smaller molecules linked together called monomers. Polymerization is the linking together of identical monomers to form larger molecules called polymers.
So when 2 monomers link together in a molecule of water is eliminated from the thing.
Reaction.
And then it's called a condensation reaction. And many condensation reactions occur when many monomers link together to form a polymer.
Condensation reactions can be reversed, so polymers broken down to monomers, by the chemical insertion of water to the polymer, and this is called hydrolysis reaction.
So, carbohydrates. Carbohydrates contain the elements carbon, hydrogen, and oxygen. Their main functions are the storage and the release of energy,
such as in starch and glycogen.
And then cellular structures, so for example the cellulose cell walls of plant cells.
There are 3 classes of carbohydrate. Monosaccharides, which are the monomers or the single units. These form the building blocks for larger carbohydrates, and the names are determined by the number of carbon atoms in the molecule.
And then you have disaccharides, which are 2 monosaccharides joined together by a glycosidic bond during a condensation reaction.
And then polysaccharides, which are large complex carbohydrates formed from very large numbers of monosaccharides joined together by glycosidic bonds.
Such as starch. Monosaccharides, the general chemical formula is CH2O and then N, so that's timesing how many carbons you have in oxygens. If N equals 3, the sugar is a triose, and the formula is going to be C3H6O3.
And then if N is 5, it's a pentose sugar, and the formula is C5H10O5.
And finally, if N equals 6, the sugar is a hexose sugar, and the formula will be C6H12O6. The most abundant monosaccharide is glucose.
And this is a hexose sugar because it has 6 carbons in the monosaccharide.
Glucose is found in 2 forms. That's alpha glucose and beta glucose. Who wants to describe the difference?
Basically, the difference is on alpha glucose. On the first carbon, the hydrogen is on the top, and the hydroxide group is on the bottom. It's pointing down,
below.
and then it's flipped on the beta glucose.
So the hydroxyl group is above carbon 1.
Mm-hmm.
And the way we count carbons is there's an oxygen, and we count that as 0. And then we go 0, 1 clockwise. So the first carbon is the first one clockwise from oxygen.
Alpha and beta glucose are isomers, so molecules with the same chemical formula, so C6H12O6, but different arrangements of their atoms.
The only difference is, as Mia said, the positioning of the hydrogen atom and the hydroxyl group on carbon 1. Other examples of hexose monosaccharides could be
fructose and galactose, and they both have the same chemical formula, C6H12O6, but they're isomers, so they have a different arrangement in their atoms.
So some other types of monosaccharides of biological importance are pentose sugars, so they have 5 carbon atoms. So for example, deoxyribose sugar found in DNA nucleotides, and triose sugars, so with 3 carbon atoms, which are intermediate in respiration and photosynthesis.
And then you have the properties of monosaccharides. So because of their small size, they're all soluble in water, and so they easily dissolve inside a cell and are easily transported in the bloodstream of animals.
Very interesting. Disaccharides are formed when 2 hexose sugars combine in a condensation reaction where a new glycosidic bond is formed with the elimination of water. So remember, when you're doing this in exams, water is produced to show that it is a product.
Condensation. Just think of the water on windows.
Okay, what are some examples of disaccharides?
So there's maltose, which can be formed when alpha glucose and alpha glucose join.
There's sucrose, which is glucose and fructose reacting.
And then you have lactose, which is where glucose will join with galactose.
And what could be the general formula for these?
C12H22 and O11 plus H2O.
And it's like that because it's lost the water.
Yeah. Okay, so maltose is found inside seeds and is an important source of glucose during germination.
Sucrose is transported through the phloem of all plants.
And lactose is found in mammalian milk and is an important source of energy for their young.
The resulting link between the 2 sugars is called a glycosidic bond.
And the bond is between carbon 1 on one of the monosaccharides and carbon 4 on the second, so it's called a 1-to-4 glycosidic bond.
Disaccharides can be broken back down to monosaccharides by a hydrolysis reaction, the breaking of the glycosidic bond with the chemical insertion of water.
Very nice.
So when you're drawing a diagram of like either the condensation reaction or the hydrolysis reaction, always label the whatever bond it is and like where water's produced.
Okay. Polysaccharides are polymers. In the case of polysaccharides, the monomers are monosaccharides, such as glucose, linked together by glycosidic bonds formed for a series of condensation reactions.
The smallest polysaccharide is made up of just 3 monosaccharide monomers linked together. However, polysaccharides are usually made up of thousands of monomers, which gives them a large size and makes them insoluble. Polysaccharides either have an energy storage or a structural function.
So glucose is the main source of energy in cells, and it must be stored in like an appropriate form. So, because glucose is soluble in water, it would affect osmosis within the cell. So, to overcome this, they convert glucose into a storage polysaccharide like starch and glycogen, which are always insoluble, so they don't affect osmosis, compact, so they can be stored in a small place in the cell, easily hydrolyzed to release the glucose for respiration, and contains a lot of energy in their bonds.
What are some examples of polysaccharides then?
So
please?
starch, which will be found in plants, and glycogen found in mammals. These consist of alpha glucose monomers and are used for storage. Whereas cellulose in plants and is it chitin or chitin?
Chitin, I'd say.
Chitin. Okay, chitin. In insects, which consist of beta glucose monomers and have a structural function.
Should we go into some more depth then? So starch, which is digested by amylase, is as she said, energy storage polysaccharide in plant cells. The starch polymer is made up of thousands of alpha glucose monomers and is made up of two different molecules, amylose and amylopectin. What is amylose?
Amylose is a straight-changed helical polymer containing alpha 1 to 4 glycosidic bonds.
This means it can be compacted, so we can store it more densely in a smaller volume. But it is slower to break down into glucose, as we can only break down one bond at a time, so one from each end.
So less energy is released.
Yeah.
Whereas amylopectin is a branched polymer, and it contains 1 to 4 alpha 1 to 4 and alpha 1 to 6 glycosidic bonds. So because it has like more branches, it has more bits to like cut off and release energy, so more energy is released.
But this does make it less easy to store, and that's why there's a mix, so we can find a balance.
And then we have glycogen, which is an animal storage polysaccharide found inside liver and muscle cells. It is made from many alpha glucose molecules and is highly branched, which allows for the quick release of glucose from the ends of branches to be used in respiration. It's very easily hydrolyzed. It has both 1 to 4 and 1 to 6 glycosidic bonds. It's similar to amylopectin with its branching, but glycogen has more branches.
And we have to use the keyword highly branched for glycogen.
Yeah.
Okay, cellulose.
So cellulose is a polysaccharide made up of beta glucose molecules. It's very important in the plant as a structural carbohydrate. It's the most important component of the plant cell wall. It gives the plant cell rigidity because the cellulose cell wall is inelastic and has a high tensile strength to prevent the plant cells from bursting.
So the cellulose forms straight change where adjacent molecules are rotated 180 degrees due to the beta glucose molecules where the hydroxyl group is, and then hydrogen bonds form between straight chains, forming cross links.
Many straight chains will be joined together by many hydrogen bonds to form a strong microfibril, and many microfibrils will form a strong cellulose bundle.
Did you mention the rotated 180 degrees?
Yes.
Chitin is in similar structure to cellulose. It differs in that some of the hydroxyl groups on each of the beta glucose molecules are replaced by nitrogen containing acetylamine. Can someone pronounce it nicely?
Acetylamine.
Acetylamine groups.
Ooh.
That's not nice.
These additions make chitin a mucopolysaccharide.
It's lightweight, waterproof, and very strong.
It forms the exoskeleton of arthropods, such as insects, arachnids, and
Crustaceans.
And it also forms the cell wall of fungi.
Very nice.
Ooh.
Okay, lipids.
Woo!
Lipids include triglycerides and phospholipids. What are they made from?
They're made from the same elements as carbohydrates, but contain proportionally less oxygen than carbohydrates. So as we mentioned before, they will be consisted of hydrogen, oxygen, and carbon.
The triglyceride.
Okay, so Lipids are non-polar. This means they're insoluble in water, since water is polar. But they do dissolve in organic solvents such as propanone and alcohols.
Triglycerides are made up of two main components, glycerol and fatty acids. So the glycerol is the backbone, and because they're made up of two different types of subunits, they are not made from identical monomers, so they are not polymers.
Because there's no repeating subunit.
Yeah.
Okay, what is glycerol?
Glycerol is an alcohol with the formula C3H8O3. It is the constant in all the triglyceride and phospholipid molecules studied.
And it contains, as Minnie said, 3 carbons linked to hydroxyl groups, and it dissolves in water, meaning it is a hydrophilic molecule. Fatty acids?
So fatty acids follow the general structure. So they're made up of 3 parts, a methyl group, which is CH3, a long hydrocarbon chain, CH2, and a carboxyl group, which is COOH. So there's many different types of fatty acids depending on the length of the hydrocarbon chain. So the higher the number, the longer the fatty acid chain. And the variation in lipid structure is due to many different types of fatty acids.
Okay, so what are the different types of fatty acids?
We have saturated and unsaturated fatty acids.
Okay, so saturated triglycerides contain saturated fatty acids, while unsaturated triglycerides contain unsaturated fatty acids.
So the hydrocarbon chain in a saturated fatty acid contains no carbon-to-carbon double bonds and has a maximum number of hydrogen bonds bound to the carbons atoms in the hydrocarbon chain. So that's why it's called saturated.
Okay, so what?
And then fats are saturated and found in animals, and they're solid at room temperature. And these have a straight chain, so they can pack closely together. That's why they're solid at room temperature, and they have strong intermolecular forces.
Okay.
And then with unsaturated fatty acids, there are one or many carbon-to-carbon double bonds in the hydrocarbon chain, meaning that the hydrocarbon chain does not contain the maximum number of hydrogen atoms, hence the term unsaturated.
And this means they cannot be closely packed, so are a more fluid lipid.
So how do triglycerides form?
Well, to form a triglyceride, one glycerol molecule combines with 3 fatty acid molecules, 1 on each carbon, and each fatty acid is linked to the glycerol by an ester bond. One triglyceride will have a total of 3 ester bonds. The ester bonds are formed by condensation reactions. So who can describe what an ester bond looks like if we drew it out?
A line.
So perhaps a different way of explaining it would be the hydroxyl on one of the carbons and the hydrogen of a hydroxyl on a fatty acid is eliminated to form water. So a C bonds to an O bonds to a C on a fatty acid.
If a lipid is broken down to fatty acids and glycerol, the reaction involved will be a hydrolysis reaction.
So a high intake of fat, especially saturated fat, damages the heart and the coronary arteries and contributes to heart disease.
Okay, so what does LDL stand for?
Low density lipoproteins. These contain and transport saturated fats and cause harm. Fatty material can block major arteries and cause a myocardial infarction.
Which is basically a heart attack, to be honest.
Very nice. So what does HDL stand for?
High density lipoproteins.
And these contain and transport unsaturated fats and carry the harmful fats away to the liver for disposal. A high proportion of HDL in the blood lowers the risk of heart disease.
So, what are some health implications that high fat intake can cause?
So it is a contributory factor to heart disease. It can lead to angina, it can lead to an aneurysm, it leads to a higher risk of cancer, and can lead to obesity which links to osteoarthritis.
Basically nothing good.
Okay, what are some properties of lipids then?
They are insoluble in water, but are soluble in organic solvents. So, for example, acetone or ethanol. Fats will be solid at room temperature. So, for example, your lard, your margarine, or your butter. Whereas oils will be liquid at room temperature. So, olive oil, for example.
And waxes contain alcohol, are hydrophobic, and melt above 45 degrees. A waxy cuticle prevents water loss.
So the functions of lipids. So the first one is energy storage, which is pretty self-explanatory to be honest. So lipids are used as an energy storage in seeds and animals because they release a lot more energy than carbohydrates, than when used as alternative respiratory substrates.
It can be used for protection of vital organs and are used as shock absorption, so prevent physical damage to internal organs, such as your liver and your kidney, which are quite exposed because there's no ribs or anything covering them.
Thermal insulation is another function, where lipids will be used as a heat or a thermal insulator. So, for example, the blubber in whales and seals reduces heat loss.
And then you have buoyancy. As lipids are less dense than water, they float and so have a role in maintaining the buoyancy in organisms like fish.
And actually, coconuts have
Coconut?
have lipids in, so they float
Oh,
because they're on
yeah.
beaches, so it can be used as transfer of seeds.
Nice.
Fun facts.
How cool.
Okay, so metabolic water is produced when lipids are oxidized by respiration. This is important in desert animals such as the camel, whose hump is actually made of fat. And then water is formed from the process of respiration.
And then finally, waterproofing. Waxes are hydrophobic and cover the exoskeleton of insects and also the cuticle of plant leaves. Both provide a waterproofing property and reduce water loss.
Very nice, very nice.
Phospholipids. Phospholipids differ in their structure to triglycerides. They have a glycerol, 2 fatty acids, and a phosphate group.
And they are important for the function of the cell membrane.
Very important.
So the phosphate group and the glycerol, they are polar. Yeah.
Making them
So
hydrophilic.
they're the polar head, so that means they're hydrophilic, so they're attracted to the water. Whereas the fatty acid tails are hydrophobic, so it means that they repel the water.
Okay, very nice.
Alright, that's it for your lipids. Now let's move on to amino acids and proteins.
Amino acids, like carbohydrates and lipids, are made up of 3 elements: carbon, hydrogen, and oxygen.
But in addition to these, they also contain the element nitrogen. Sulfur is also found in some amino acids, such as cysteine.
Sorry. So the structure of amino acids. So they have the amine group. You have a carboxyl group, which are opposite each other around a carbon, and then you have you just have a single hydrogen, and then you have your variable R group. So the R group is called the variable group and represents the rest of the molecule, and there's 20 different variable groups. Yeah.
Which form 20 different amino
Which
acids.
form 20 different amino acids. That's what I was looking for.
Dipeptides.
2 amino acids can be joined together to form a dipeptide. You can gather this with di meaning 2. They will combine in a condensation reaction involving the amino, so the NH2 group of one amino acid, and the carboxyl COOH group of another.
So the resulting link is called a peptide bond, and the dipeptides can be broken back down to the amino acids by hydrolysis reactions. And there's no oxygen in the bonds like carbohydrates and lipids, which like there usually is.
So polypeptides are formed by many amino acids being joined together by peptide bonds. Polypeptides are polymers, where amino acids are the monomers, because they only have small differences in, so we can count them as repeating subunits.
Yeah.
So amino
acids
form polypeptides or chains of amino acids, and polypeptides will form proteins. All proteins have a primary structure and a secondary structure. Sometimes proteins will have a tertiary structure, and occasionally proteins will have a quaternary structure.
So the primary structure of a polypeptide refers to the number and the sequence of amino acids in a polypeptide chain. And there's variety in the different proteins found in nature because there's 20 different amino acids, and they can have any number and sequence of amino acid in a polypeptide chain.
So, secondary structure. So the primary structure of the polypeptide can coil to form either an alpha helix or a beta pleated sheet. So these are types of secondary structure.
The most common type of secondary structure is the alpha helix, and this is held in a spiral shape by hydrogen bonds between the oxygen on carboxyl groups and the hydrogen on the amine groups.
So some fibrous proteins, which basically means they don't fold any further than the secondary structure, have an alpha helix linked into rope-like strands. So keratin and collagen. So keratin is found in hair and nails, and collagen is found in the connective tissue and skin. A less common secondary structure, also with hydrogen bonds, is the beta pleated sheet, which is a flat zigzag structure. So the protein silk.
Ooh. Tertiary structure.
So these proteins are more complex. The secondary structure basically folds to give complex three-dimensional shapes. So these are globular proteins. So for example, your enzymes, your hormones, plasma and cell membrane proteins. So the unique tertiary structure will determine its function.
This can be formed by hydrophobic interactions on the R group away from the water. The secondary structure is folded and is held in a specific 3D shape by bonds or interactions that form between these R groups on some of the amino acids in the polypeptide chain. What are some other ways we can bond on a tertiary structure?
So
Do you
we've
wanna name one
already heard of
each?
hydrogen bonds, which form between your oxygen and your OH.
And then you have ionic bonds, which form between oppositely charged R groups.
You can have disulfide bridges, which form between sulfur atoms in like cysteine.
Hydrophobic R group interactions, which will occur on the inside of the protein, so away from water.
And hydrophilic R group interactions on the outside of the protein, so where it's polar and fun.
Okay. The quaternary structure occurs when more than one polypeptide chain, each with a tertiary structure, combines to form a larger protein complex.
The tertiary structures are joined together by bonds similar to those within the tertiary structures themselves, so ionic, hydrogen, disulfide, and hydrophobic interactions. Quaternary proteins are sometimes associated with non-protein groups called prosthetic groups. So hemoglobin is a relevant example, which is where a protein carries oxygen in mammalian blood. It's made of 4 polypeptide chains, each with a tertiary structure. It also has non-protein components, or prosthetic groups, called heme groups, containing iron.
And that's where the oxygen binds to in the hemoglobin.
Okay, so how can we classify these proteins? We can classify them according to their structure. So one type would be
Fibrous proteins, which is keratin and collagen, which can carry out a structural function. So collagen is made up of 3 alpha helix polypeptide chains twisted into rope-like strands, which are held together by hydrogen bonds. So some of the properties, they're tough because they have lots of hydrogen bonds. They're non-specific and they're insoluble. And they're long and thin and strong.
And then we also have globular proteins mentioned previously, which can be tertiary proteins. So for example, your enzymes and your plasma proteins, or quaternary proteins. So for example, hemoglobin and antibodies. Properties include being compact and folded into spherical molecules. They will have a
Specific.
specific shape and are soluble in water, making them easily transported through the bloodstream, for example.
Okie dokie.
Biochemical food test!
Okay, so one biochemical food test would be for reducing sugars, such as glucose. To your test tube, you would add Benedict's reagent and your test sample. Then what would you?
And then you'd boil it in water, and if you have a positive result, it would go from light blue to brick red upon heating.
Whereas a negative result would be the light blue colour the Benedict's reagent would stay. It would stay light blue upon heating, which means non-reducing sugars such as sucrose may be present.
Samples of reducing sugars may give a result with Benedict's reagent that is graduated between green, yellow, orange, or brick red. The more concentrated the reducing sugar, the nearer brick red the colour will be. This is semi-quantitative.
Semi-quantitative meaning that it will only give you a range, not a specific value. So
Then you have non-reducing sugars, so sucrose. So if you get a negative result in the Benedict's test, you can add hydrochloric acid to the test sample and boil it. So that will hydrolyse any glycosidic bonds that are present. And then if you add hydrogen carbonate powder, which is an alkali, to neutralise it, and add Benedict's reagent and boil, if it's a positive result, you'll get a light blue to brick red / orange. So this shows it was in the sucrose was in the original test. And if there's no colour change, that means you've got a negative result. So no reducing or non-reducing sugars are present in the sample.
Okay, another biochemical food test you need to know is the one for starch, where after you add iodine solution, it will turn from an orange-brown colour to a blue-black colour when starch is present. This is a qualitative test and an accurate concentration cannot be determined.
Another test would be for proteins, and this is for soluble proteins and is called the Biuret test. When a few drops of Biuret reagent are added to a protein solution and gently shaken, the solution will turn from a light blue into a purple colour. If the sample solution does not contain protein, then the blue Biuret reagent will stay blue, no colour change is observed, and remember, no heating is needed for the Biuret test.
And then finally you have your lipids test. So lipids are described as being hydrophobic, so they repel water, and will only dissolve in organic solvents like ethanol. So if you mix the test sample with an equal volume of absolute ethanol to dissolve in any lipids, and then you add an equal volume of water and shake gently, the lipid will form any small droplets and appear as a white and cloudy emulsion.
Remember to use both these descriptive words. Okay, so
water, H2O, will consist of two hydrogen atoms covalently bonded to one oxygen. Water is a polar molecule as it has an uneven distribution of charges. So the oxygen end of the molecule will have a slight negative charge and the hydrogen ends have a slight positive charge. So when two water molecules are in close contact, their opposing charges will attract each other, forming a weak hydrogen bond. So many of the properties of water are due to its ability to form hydrogen bonds.
Individual hydrogen bonds are weak, but many of them form a strong lattice framework. Water molecules tend to stick together as a result of formation of hydrogen bonds. This is known as a cohesion.
Okay, so why is it useful in plants in the xylem vessel? This is because it allows water to stick together in a column in the xylem and be drawn up a tree against gravity in one unbroken column in the transpiration stream. Some properties of water would be
surface tension, which is where water meets air, and the water molecules form hydrogen bonds with other nearby water molecules, but not with the water molecules in the air. So this cohesion produces an uneven distribution of force called surface tension, and the surface of the water forms a skin which can support aquatic plants and insects such as pond skater.
And it can even support animals on the underside of it, so some can stick to the bottom of it.
Property number 2, water is a universal solvent due to its it being a polar molecule. So it can attract and dissolve other charged particles such as ions and other polar molecules like glucose. Its property as a universal solvent makes it an excellent transport medium. So in animals, blood will transport dissolved substances around the body, whereas in plants, xylem vessels will transport water and dissolved mineral ions.
Why don't lipids dissolve in water?
Because they're non-polar molecules, so they repel water.
Water is also a metabolite. This means it's a reactant in photosynthesis and hydrolysis. It's also produced during aerobic respiration and condensation reactions.
Water also has a high specific heat capacity, so this is because of a lot of energy needed to break the hydrogen bonds and increase the temperature of water. So it provides a more stable environment for aquatic organisms as it prevents dramatic changes in temperature, and it also allows enzymes to efficiently catalyse important chemical reactions at the temperature inside the body without becoming denatured.
Very nice.
It also has a high latent heat capacity, so also known as latent heat of vaporization. And this is because a lot of heat energy is needed to break the hydrogen bonds and change water from a liquid to a vapor. So meaning that a lot of heat energy is required for water to evaporate from the surface of an organism, providing a significant cooling effect as heat is removed from the body to evaporate the water molecules, so sweating in animals or transpiration in plants.
Water is also less dense in its solid state compared to liquid water. Ice therefore floats on the surface of water, forming an insulating layer which allows aquatic organisms to survive in water beneath the ice. Also provides a habitat for organisms such as polar bears on top of the ice.
And finally, water is transparent, this allows light to pass through the underwater aquatic plants, and enables them to photosynthesize effectively.
And that, my friends, is it.